Hi, this is Mr. Wartsky, and this is the second in the Meiosis screencast series. Remember, if you have any questions, to either review the video, read your textbook, or ask me the next day in class. So we have finished Meiosis 1. We are going to now start off with Meiosis 2. And Meiosis 2, which is what M2, that's I2, it's interphase 2. So in interphase 2, meiosis 2, I'm sorry, is the division of the chromosomes. Interphase 2, there's G1, there's G2, but there is no S, and there is no S. Make sure that you understand that there is no DNA synthesis. I think I've included enough exclamation points to emphasize the fact that there is no S. Draw the pictures of two cells. See, we have one cell, two cells. Now draw two cells. Draw two cells. Beautiful. So meiosis 2 prophase 2 is just like mitotic prophase. Chromosomes condense because of MPF. The nuclear membrane fragments because of MPF. The nucleolus disappears because the chromosomes condense. Spindle fibers form. So now we have two cells. Each cell has two chromosomes made up of four chromatids. So we have one cell, two cell. We're going to draw two cells now because that's what we have. And each cell has two chromosomes, and we have a total of four chromatids. Now, the purpose of meiosis 2 is to divide those chromosomes so that each one is made up of a single chromatid. In metaphase 2 of meiosis 2, of M2, the kinetochore spindle fibers lengthen and shorten so that the kinetochore lines up on the metaphase plate, and that is a checkpoint. APC is produced. Again, that should sound familiar because we've been saying the same thing over and over again. In this case, we have still two chromosomes, and we still have four chromatids. Draw the picture. This is metaphase 2. Hit pause if you need to. In anaphase 2 of meiosis 2, the APC, anaphase promoting complex, will break down the kinetochore. The kinetochore spindle fibers will shorten, and the chromatids will separate. And here's a picture. Now we start to see that each cell is going to have two chromosomes, which is half of four, and each chromosome is going to be made up of one chromatid. Each chromosome is going to be made up of one chromatid. Please draw that picture. In telophase 2, chromosomes decondense. The nuclear envelope reappears. The nucleolus reappears. The spindle fibers become disorganized, and we can start cytokinesis. That should sound familiar again. Now, in here, we're going to see that each cell, right? We're going to have 1, 2, 3, 4 cells. Each cell is going to have two chromosomes, and each chromosome is made of one chromatid. So you're going to have two chromosomes and two chromatids. So make sure you draw that picture there. And each cell, notice, is unique. Each cell is different as far as the chromosomes go. And that's huge. That provides variation. That's so big. Cell divides in cytokinesis 2 of meiosis 2. There are now 4 unique gametes. And each gamete has half the number of chromosomes. And each gamete will be made up of single chromatids. So we started off with 4 chromosomes made up of 8 chromatids. Now we're ending up with 2 chromosomes made up of 2 chromatids. Amazing. Draw the picture. So meiosis in you people, males. So in males, our testes are made up of a long seminiferous tubule, so about 375 feet per testes, like a long wound up string, like ball of yarn or ball of string. In the walls of the seminiferous tubules are cells known as spermatogonia. And when they start meiosis 1, they will become primary spermatocytes. They finish meiosis 1. They begin meiosis 2. They will become secondary spermatocytes. And after the end of meiosis 2, they will become spermatids and then they will be reorganized to form sperm. So we start off with one spermatogonia, and you'll end up with 4 spermatids, which will become sperm. And here's a diagram of it. This is spermatogenesis. This is spermatogonia. They start with primary spermatocyte, which becomes a secondary spermatocyte, forms the spermatids, which will become sperm. All right, so this will be the primary spermatocyte, secondary spermatocyte, spermatids, sperm. This is the male. Don't look at the female yet. We'll get there. Okay, male. Males are easy. One cell becomes 4, half the number of chromosomes, 1 chromatid per chromosome. We're good. Move on. For individuals, there are anywhere from 250,000 to 1 million eggs per ovary. We're going to say 250,000. That means that each individual has 500,000 eggs because you have 2 ovaries. All of those things are the primary oogonia. Okay? So each cell is a primary oogonia, and they will undergo meiosis to become the egg. So they all become primary oocytes at the same time. They start meiosis 1 before you're even born. You go through prophase 1, you cross over, and then they stop. The eggs stop and they stay in stasis. There's no more sex at all. All the sex happens before you're born in all 500,000 primary oogonia. Now, when you start meiosis 1, it is called a primary oocyte. Okay, so now you have 500,000 primary oocytes stuck right after crossing over, and then you're born, and you grow up, and then you have the change. You know, the change. You see movies about the change. You talk about the change. Guys don't go through that stuff at all. We don't talk about these things. We learn from the street. But the change, what happens in the change? Well, a set of eggs, because of hormones, will start to develop. The largest one produces the most estrogen and will stop the smaller eggs from developing. The largest of the primary oocytes is ejected from the ovary once a month, and it will finish meiosis 1 when it's ejected. But when it goes through meiosis 1, there is an unequal cytokinesis. So it does not divide equally. You have one big plump cell, and that's a primary oocyte, and the other one is a really tiny cell. It's basically a chromosomal trash can. That's called the primary polar body, and eventually the primary polar body will die. The primary oocyte enters the oviduct and is moved towards the uterus, and it will start meiosis 2, and it will stop after prophase 2. Okay, so it starts at meiosis 2, so it's a secondary oocyte, and it'll stop after prophase 2. And if you're not fertilized, this secondary oocyte will be lost in menses. However, the sperm, if it meets the secondary oocyte, once the sperm fertilizes the secondary oocyte, it will start to go through meiosis 2. It'll finish meiosis 2. And then again, you will have unequal cell division. Unequal cell division, you'll form the secondary polar body, which is a chromosomal trash can, and you'll form that nice plump egg. Nice plump egg, and that plump egg is called the ovum, and that will become junior. So the DNA from the sperm will meet up from the DNA from the ovum, and we will now have a diploid organism, which will become the baby. It will continue down the oviduct for 3 to 5 days, and it will become a ball of cells called a blastocyst, which will implant into the uterine wall when the uterus is at its plumpiest and fluffiest, like a pillow. So that's right here. We have the primary oocyte. It finishes meiosis 1. We have a secondary oocyte. Polar body dies. That doesn't go any further from here. I don't even know why this does this here. We have the secondary oocyte starts meiosis 2. If it's fertilized, it will finish and become the egg cell here. Secondary polar body dies. This right here, when the two sets of chromosomes line up, it's going to be the ovum. This is going to be junior right here. This is baby. This will divide and divide and divide, form about 100 cells, become a blastocyst, and implant into the uterine wall, the fluffy uterine wall. So what do we have? Problems with meiosis. Well, there can be an unequal exchange of DNA during crossing over. This can act as a deletion or an insertion. If there's a deletion of one DNA strand, it's going to be an insertion into another DNA strand. We can have an inversion where the piece of the chromosome that's being exchanged can flip and then join and fuse, so the whole section of DNA is flipped around. Or you can have a translocation in which the piece of the chromosome that's being exchanged is lost. It floats somewhere else and joins to a non-homologous chromosome. That's a thing. Or you can have non-disjunction, where chromosomes don't divide properly during anaphase 1 or anaphase 2. The APC doesn't do its job. The kinetochore spindle fibers don't shorten or attach to the synaptonemal complex of the kinetochore. So the resulting gametes will have the wrong number of chromosomes, and that is called aneuploidy. Aneuploidy could be 2n plus 1, which means that's 46 plus 1, that's 47. It could be 2n minus 1, which is 45. Right? Or polyploidy, in which a whole set of chromosomes can move at the same time. So you can have 3n, which is a triploid, or 4n, which is a tetraploid. Now, polyploidy doesn't happen in human people or mammals or animals that much. It can only happen in plants and maybe some insects, and that's about it. Don't get too carried away with too many chromosomes, because too much of a good thing, except for toilet paper and love, is bad. And here's some examples of how non-disjunction can occur. This is normal. This is when you have problems. So for here, 2 chromosomes move over to this side, and we get 50% or 100% of the gametes here are affected. Whereas over here, if it happens in meiosis 2, only 50% of the gametes will be affected. Might want to draw these two. Pause it. And that's it. Oh my gosh. So we are done with meiosis. Yay. If you have any questions, please review the video, or you can look in your textbook, or you can talk to me tomorrow. Have a good night.